I want to start showing you this picture, which was taken many years ago in Japan, because the event that led to this tragedy was an eye-opener. Here's a mother bathing her mentally retarded son, and you can see that son is very, very sick. He was poisoned by mercury, by methylmercury, from the mother's diet during pregnancy. So the mother was unscathed. She escaped from the poisoning, but the methylmercury made it through to her fetus during her pregnancy and destroyed that nervous system forever. It was an eye-opener for us because it illustrated that chemical brain drain is a reality. The big question is, how large a problem do we have in front of us here? And that's what I'm going to try to determine. Now, we have done much research in this field, but we have made some erroneous assumptions. And those erroneous assumptions have negatively influenced on our capability of reaching the right conclusions. First of all, we thought that when a woman is pregnant, the child is in the uterus, the fetus is protected by that armor that the mother has wrapped around this developing child. Wrong. We thought that children are just little adults because basically, they have the same organ systems, the same, in a different scale, the same anatomy. Wrong. There are definite changes in the physiology. We also thought that any poisoning that might occur would be reversible if it happens, just like infections and other childhood diseases. Not so. And we thought that industrial chemicals are so useful to us that we need proof before we limit their uses. And unfortunately, the kinds of tests that can be carried out to assess the toxicity of industrial chemicals, they are too expensive. So this is like a double whammy that we really want the proof, but we can't afford to look for it. And for all of these reasons, we have ended up being in serious error in regard to the impact of chemical brain drain. I want to tell you a little bit about brain anatomy and physiology because the reasons that I've listed here in this slide indicate that the brain is an extremely complex organ that has to undergo a variety of developmental stages in a particular sequence, and they must happen at a particular time. If that does not happen, that's it. You only get one chance to develop a brain. So if we want to do research in this field, figure out how this jigsaw puzzle is put in shape and how we develop our brains for use in the rest of our lives, whether we watch television or computer screens or whatever, we need some tools, some methods to examine brain functions and brain development, and we need to understand what pollutants, what environmental chemicals can actually do to inhibit normal development of the brain. We have chosen to conduct studies in a community in the Faroe Islands. You can see here is this map that NASA has taken from space, and see Scotland over there on the left, and the Faroe Islands on the upper left corner of this picture. That's about 50,000 people living on these islands, and they are a fairly homogeneous society, and they eat a lot of fish. It's a fishing community, and for that reason, they are actually exposed to some toxic agents that are concentrated in the food chains in the oceans. So we have conducted studies in the Faroes, and we have collected blood from births, from the umbilical cord, and analyzed it for, in particular, mercury. And I'll show you another slide from the Faroes here. They have this ancient tradition of catching pilot whales, and those whales accumulate mercury, and therefore the population has an increased exposure to this substance. Now, after we had collected bloods from all of these kids in the Faroes, we then started conducting medical examinations. We called the kids in with their mothers, and we used a variety of neurophysiological methods to examine whether any changes had happened. And this slide here shows my Japanese colleague, Professor Murata, getting ready to examine a child. And he's actually trying to measure how fast nerve cells are communicating with each other. That is, the speed of the electrical signals from one relay station to the next. I'll show you some of our results. The exposure scale is the horizontal line, and then the time that it takes for the signal to jump from one relay station to the next is on the vertical scale. And the surprising discovery we made is that even at the fairly low exposures in this group of kids, we could still see that there was a delay in the conduction speed. And you can also see the US EPA exposure limit is sort of way up there. And we can detect delays in the brain that occur at exposures that are very clearly below the exposure limit that we thought would protect people from toxic effects of methylmercury. We also use some other more sophisticated techniques to look at how the brain functions in kids exposed to methylmercury. You can see on this magnetic resonance imaging picture on the right, you can see the brain structure. And what we've done is to ask the child to do this finger tapping with the left hand. And the nerves cross from one side to the other in the spine, and so the motor cortex here on the right-hand side should light up, and it does. So when you do this with your left hand, the motor cortex on the right lights up in the kids who are not exposed to high levels of mercury. But if you look at these kids here, this is an average picture, you can see that at high exposure, they actually have to use both sides of the brain. This is very clearly abnormal. And I would think that it's an indication of immaturity of the brain, that the brain hasn't quite decided which side to use when controlling the left hand or left leg or whatever it is. It's a sign of immaturity. At least it indicates that the nerve cells have not found their optimal way of communicating. We also used some neuropsychological tests to see how good the kids were at remembering numbers and words, and how good they were at spatial discrimination and all sorts of functions like that. The general finding was that every time the mercury dose doubled, the child lost functions that compared to about maybe 1.5 or 2 IQ points. So essentially, well, I mean, most of us have average IQ levels that are at least 100, and with at least 100, so what does the loss of a couple of IQ points mean to us in practice? But I've never met a person who was willing to give away or donate any of his or her IQ points simply because the brain is such an essential organ. The brain is what makes us who we are. It's what makes us humans. It makes us individuals. And therefore, every single part of that brain function that we have is essential to us. So that is what's different from, let's say, kidney function. Because you can donate one kidney to, let's say, it's a sibling who needs a transplantation, and you can function well with that single kidney you have left. Not so with the brain. We also found that when we re-examined these children later on, that the functions had not improved. We continued to see the same delays. So this means that once you have received some sort of chemical brain drain, some damage early on during development, that stays with you for the rest of your life. So let me illustrate this with a curve. Now, this shows you the distribution of IQ points in the population. And you can see on the far right, the 2.5% geniuses who have very high IQ levels, like you. And then on the other extreme, on the left, you can see those who are really in trouble because they don't function very well. Now, if you expose that population, essentially prenatally or in early development, to some poison that shifts the average toward the left, so that the average is only 95 instead of 100, you can see that now you suddenly have 4% that are mentally retarded. And so it's a huge increase, relatively speaking, in those who are in difficulty and who need special education in school and all sorts of support. And if you then look at on the right, you can see that the number of very smart people has decreased similarly. So essentially, you could say the people in the middle, they are more or less unchanged, but you have shifted some of the very smart people to some who are really functioning very poorly. So we have studied mercury for 25 years, and so this is just one substance. And we've also looked at lead, and we've looked at arsenic and PCBs. But the big question is, is this just something that relates to some individual specific substances like lead or methylmercury? So we looked at the world literature on poisonings to see what are the chemical substances that can cause brain symptoms, toxicity within the brain. So this could be from poisonings or industrial accidents, from suicidal attempts, various other occasions where people have been exposed to chemical agents, and we wanted to see if the chemicals can penetrate the blood-brain barrier, get into the brain, and cause toxicity to the brain cells. We found that there were 201 industrial chemicals that are known to be brain drainers. Now, the biggest group were the pesticides. And obviously, pesticides are generated in order to be toxic, and oftentimes to be toxic to insect nervous systems. And our nervous system is very similar biologically, biochemically speaking, to what insects have. So it's no wonder if something is toxic to an insect brain, it can also be toxic to the biochemistry that we have between our ears. But there's also another group of metals, and of course lead and mercury are there. There's solvents, and then there's a mixed bag of other industrial chemicals. But the surprising thing was that about half of these substances are high production volume chemicals. So these are the substances that we use the most in society. And they probably also occur quite often in environmental pollution. So we have an iceberg here with a chemical universe of about 100,000 chemicals. And about that iceberg, we only know a little bit about 1,000 of these substances. We know that they are toxic to the nervous system in experimental animals like rats and mice. Then we have the 200 that are documented clinically in medicine to be toxic to the brain, usually the adult brain. And there's this little tip of the iceberg here. Those are the substances that are toxic to the developing human brain. There's only 5: methylmercury, lead, PCBs, arsenic, and toluene. Toluene is a solvent, and for those 5, we know for sure that they're toxic to the developing brain. And we're now beginning to regulate exposures to these substances to protect the human brain during development. But we are not doing that in regard to the 200 that we know are toxic to brains in general, and we're not doing very much in regard to the rest of the chemical universe. We are not testing industrial chemicals for neurotoxicity. That seems to be a strange way of preventing the human population against neurotoxicity, because the experience is that in science, you can see there's this horizontal scale, the time scale. There's this line that moves upwards. It starts with recognition of neurotoxicity in adults, or it could be occupational exposures. And then we start from getting more information from poisoning episodes, and we do some targeted examinations of perhaps child populations or pregnant women or whatever it is. And eventually we find that there is a silent pandemic going on, because these kids who suffer from chemical brain drain will not necessarily have a diagnosis. They may just be a little more slow. They may have some behavioral derangements of various kinds, and they may have, not be doing so well in school as they should have. So, and you can see that lead has moved all the way. It followed that curve from the very beginning. So lead is on the very top on the right-hand side, and methylmercury is right behind. And then there are some other toxicants that are perhaps on their way to move up along this line. So what do we do in this situation? We can see this pattern developing, and we think that neurotoxicity, when it happens to the developing brain, could be quite disastrous. Well, Here's what we did in regard to lead. So let's see what we can learn from that history. The various indications here from 1960, that's the safe level of lead concentration in the blood in a child. And you can see that that decreased with time. And actually now there's a committee in the United States that has just recommended that the safe level should be cut by 50%. But there's another committee that has recently said we need to go even further down because we know so much about chemical brain drain by now that we ought to go all the way to protect human brains as much as we can. But this also means that in the 1960s, for example, when we were so permissive that we accepted up to 600 micrograms per liter, we also accepted that children could be exposed to such high levels that we today are shocked that that was actually considered acceptable and normal. There are big stakes here because we can see that the prevalence of ADHD, autism spectrum disorders, all sorts of problems, behavioral abnormalities, need for special education in school, we can see that they are increasing in frequency. So 1 out of 6 children have some sort of neurodevelopmental abnormality. It's a big problem, but we don't know the exact causes. We can also see that lead exposure, mercury exposure, other exposures are so prevalent that if we calculate the costs in terms of lost lifetime earnings, we're really talking about billions of dollars per year. And this is for the United States, but the numbers are similar for the European Union. So we have a big problem in our hands, and the question is, what should we do? I have some suggestions for what I think are the prudent conclusions, at least some that we need to consider to counterbalance the erroneous assumptions that I began talking about a few minutes ago. So, number one, we get only one chance to develop a brain. And the development has to go through multiple stages, and it's so complex that just a little bit going wrong can have severe and permanent effects. The brain is very vulnerable, and it is not forgiving in regard to the effects of even small damages. We depend on the full integrity of the brain for optimal functioning. Therefore, we need to protect every bit of it. We need all the brain cells that we can have. And it's not just a matter of looking for neurological diagnosis. This is a matter of functioning. It's a matter of our own functioning, but in particular, it's a matter of the next generation's brain functions. So it's a matter of intelligence for the future. So for all of these reasons, I would say brains need protection. We need to be more precautionary in the way we deal with chemical agents in society. The next generation deserves it. Thank you.